We always hear about DNA as the code of life, right? But have you ever stopped to think about it as, well, a brilliant piece of engineering? I mean, evolution had to solve a massive problem. How do you store and protect the most valuable information in the universe? Okay, let's dive into this absolute masterclass in molecular design. So, for this molecule to actually work, it had to overcome two epic challenges. First, it needed to be incredibly stable, right? I mean, it has to survive for an entire lifetime without falling apart. And second, it needed what we call absolute specificity, a way to write and copy information with perfect, flawless precision. So how on earth did life solve both? All right, let's tackle that first challenge head on: stability. How did nature build a molecular chain that's strong enough to carry your entire genetic blueprint every single day without breaking? Well, the answer starts with a really simple but elegant idea. The very first step was to create a protective scaffold. The really precious stuff, the genetic bases, are attached to this constant repeating backbone. You can kind of think of it like the sturdy frame of a building. It provides all the structure and shields the really delicate code that it's carrying. And the real hero of this structural frame is a special chemical link called the phosphodiester bond. Now, this isn't just any old connection. We're talking about an incredibly strong covalent bridge that locks each piece of the chain to the next. That's what creates this continuous, super resilient polymer. And how it forms is just beautifully simple. It's basically a two-step process. First, you've got a phosphate group that gets ready to link up two sugar molecules. Then a water molecule gets kicked out and, boom, a new unbreakable link is forged. This just keeps happening over and over, building up that tough-as-nails sugar-phosphate backbone. But here's where the engineering gets really, really clever. This backbone isn't just the passive shield, just sitting there. It has its own built-in defense system. It's actually an active defender of the genetic code. And this shows you exactly how. See, each one of those phosphodiester bonds carries a net negative charge. This charge creates something like an invisible force field that actively repels other negatively charged molecules, chemical attackers that would love to come in and just slice through the chain. So it's a self-defending molecule. It's just an amazing way to make sure the code stays safe for decades. Okay, so life built an unbreakable chain. Challenge 1, solved. But a strong chain is totally useless if the message it's carrying is just a bunch of gibberish. And that brings us to the second huge engineering problem: how to create a code with absolute specificity. So the question now is, how do you actually write information onto this super strong backbone? See, DNA stores its code using two chains that pair up. But for this to work, the pairing can't be random. It has to be perfectly predictable, perfectly replicable every single time. There had to be a rule book. And the first big clue came in 1950 from a biochemist named Chargaff. He was analyzing the DNA from all sorts of different species, and he found this bizarrely consistent pattern. The amount of one base, adenine, or A, was always almost exactly the same as the amount of thymine, or T, and the exact same thing was true for guanine, G, and cytosine, C. And you can see that rule in the hard data right here. Look at human DNA. A and T, they're both right around 30%. G and C are both hovering around 20%. Now look at something completely different, like E. coli bacteria. The overall percentages are different, sure, but the rule, it still holds. A always equals T, and G always equals C. This was some kind of universal law, but why? Well, the reason for this pattern isn't some crazy coincidence. Turns out it's a direct consequence of the actual physical shape of the molecules themselves. The answer to the puzzle wasn't about the quantity, it was all about the chemistry. And here it is, the crucial piece of the puzzle. The shapes are literally designed to fit together like a lock and key. Adenine and thymine, they're perfectly shaped to form two nice, stable hydrogen bonds between them. Guanine and cytosine, they're shaped to form three even stronger hydrogen bonds. So A won't fit with G or C, it only fits with T. And that right there is the fundamental rulebook for life's code. So there we have it. We've got our two solutions: an unbreakable, self-defending backbone for stability and a super precise set of pairing rules for specificity. So what happens when you put them together? You get one of the most iconic, most beautiful structures in all of science. This is the grand synthesis. Those two negatively charged backbones form the protective outer rails of this twisting ladder, and on the inside, the specific AT and GC pairs form the rungs, holding the two strands together with perfect fidelity. You've got stability on the outside and specificity on the inside. It's just perfect. When you step back and look at it, what you have is a molecule that is perfectly engineered for its job. Every single feature, those strong phosphodiester bonds, that repulsive negative charge, the complementary shapes of the bases, every bit of it has a purpose. It really is the ultimate solution to the problem of storing life's blueprint. It's just an amazing reminder that biology is absolutely filled with this kind of elegant, purposeful design. It really makes you wonder, doesn't it? What other incredible engineering solutions are still out there, just waiting for us to discover them, hiding in the code of life?